Can somebody confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, let me share my screen. I'll share. Should be seeing my screen now. Today is August 14th, and we should be finishing chapter 8 and beginning chapter 13. I don't know if we'll get that far today. In the lab today, there is no lab. Work on your unknown project, and your infectious disease paper is due this Saturday. Any questions about anything?
I have a question.
Uh-huh.
If I submitted for another test on the unknown project, when will I get the next one? Is it always going to be on the weekend?
No, it will be dependent on the number of days that that test needs to incubate. So you have to
Oh, okay.
what test you ask for. If you ask for a test where you get the results immediately, you might get the answer the same day you ask. But that would only be if I check after you ask for the test.
Oh, I
Most of the
see.
tests take 2 days, so they have to incubate for 2 days.
Okay, I think I did the methyl red.
That one will take 5 days incubation.
Okay.
And you should look that up when you ask for a test. You should go read about the test. And you can
Yeah,
find
I knew
it
it
on my
was the
webpage.
5. I knew it was the 5 days, but I just didn't know if you waited till like the weekend or if it was just the number of days. So that's why I just was
It
trying
has
to
to be
clarify.
a number of days after the time it incubates. It may also be, I don't send out test results every day. And if you request a test like on Friday, I mentioned this in the lab at the start of the unknown project, and I've already sent out the results for that Friday, then I'm not going to look at it probably until Monday. So the earliest you'll get the results would be Monday. Now, occasionally on the weekend, I may send out results on Saturday, but I'll never send out results on Sunday.
Okay,
Okay.
thank you.
All right. So I had that we had talked about mutations and we discussed the base substitution and the nonsense mutation. Let's talk a little bit more about a frameshift mutation. That's when you insert or delete one or more bases. And where you, let's see, we're here deleting an A there. So you will change phenylalanine because instead of it being AAA coding for UUU, you will then remove that third U and then you pull the G from the glycine because the codon is always 3 nucleotides, and that changes that amino acid. It also changes potentially all other amino acids because the reading frame is changed. Instead of having GGC, it now becomes, sorry, I'm a little lost here, GCU, meaning it pulls that U from the stop codon. And after the addition or the deletion of one base, like I said, you could change the entire reading frame of the codons after that one change. And that's why it's called the frameshift mutation. A frameshift mutation could be more than one base, but understand if you remove or add 3 bases, you change the one base where you have the change. Let's say we have AAA, and then we remove all 3 of those AAAs. You'll then have methionine, lysine, no longer phenylalanine, but glycine will remain the same. And then after the change, the codon's reading frame is the same. So if you have a multiple addition or a multiple deletion of 3 or a multiple of 3, it is not a frame shift mutation. Any question about any of that? All right. As I mentioned, the causes of mutation is you can have a spontaneous mutation, and that's simply when the DNA is being replicated, a mistake gets made, and that we call a spontaneous mutation. You can also have a mutation caused by a mutagen, which causes the mutation. There's two types of mutagens, radiation and chemical mutagens. There are two types of radiation which can cause mutations. There's ionizing radiation, and these are radiation with a high energy like X-rays and gamma rays. And when they hit an electron, it gives that electron enough energy to escape the atom. When you lose an electron from an atom, you then form an ion. Well, it's called ionizing radiation. That ion can then react with DNA, mutating the DNA. We're not going to talk about repair. The other form of radiation that can cause a mutation is non-ionizing radiation, but specifically UV radiation. Like visible light is non-ionizing radiation, but visible light cannot cause a mutation. UV radiation can. When UV radiation strikes DNA, let me blow this up. If there are two thymines next to each other, the UV radiation can cause the thymines to form a dimer between the two thymines. The thymines are not connected with two covalent chemical bonds. When the thymine forms a dimer, meaning it has two covalent bonds binding them after UV radiation, the thymines will not hydrogen bonds with their corresponding nucleotide, in this case A. That will cause a bubble in the DNA, and this bubble cannot be replicated, meaning the DNA polymerase cannot replicate this, and then it cannot be transcribed. The RNA polymerase cannot move past the thymine dimer as well. We're not going to talk about the repair, but light repair mechanisms can repair thymine dimers. Any question about any of that? All right, let's talk about chemical mutagens. We're only going to talk about one example, nitrous acid. It is a chemical mutagen. When nitrous acid, shown here, reacts with DNA, it actually chemically modifies the A. It's still an A, but it's a changed A, and that A does not act like an adenine. The A acts like a guanine, so when the DNA polymerase sees this modified A, it won't read it as an A, it'll read it as a guanine, and then it will insert the C opposite the A. When DNA replication happens again, this mutation will then be fixed, meaning this C will be replaced with a G. And that we call, well, almost an inversible reaction. While the right here actually is where it has to happen, because if you get it repaired here and you remove the A, you'll fill in a G there, so that won't repair it. But if you notice that the DNA is mistaken, meaning that that A isn't quite acting like an A, you can actually repair the DNA there. The problem is repair normally happens on the newly synthesized strand. And if that's the older strand of DNA, then actually that T will be removed and it'll be put in with a C because that A will be read like a G. Anyways, that's how nitrous oxide acts as a mutagen. It changes the adenine, so adenine doesn't act like an A, it acts like a G, and then the mutation will occur. Any questions about chemical mutagens? The frequency of a mutation is for each, oops, trying to move that out of here. For each 10 to the 9th replicated basis, the DNA polymerase makes one mistake, meaning one base out of 10 to the 9th, that's a billion nucleotides, gets the wrong nucleotide put in the newly synthesized strand. Now, because a gene has about 1,000 bases in the gene, that makes the mutation rate one mutated gene in 10 to the 6 genes. And you'd say that's not a very big number, and spontaneous mutation is not very frequently. But if you remember, the human population is close to 1 times 10 to the 8th. It's a little less than that, I think. I haven't looked recently, but it was getting close to 1 times 10 to the 8th. And that means every year we have several children born on the planet which are actually mutants in one gene of interest. I don't know what the number of children, babies born in the country, not the country, the world is, but I bet it's over 1 million. Because the population is 1 times 10 to the 8th, and 1 million is 1 times 10 to the 6th. And my point is that for any gene we look at in the human population, we're getting every year babies born which are mutated in each gene of interest. Any question about any of that? Now, mutagens increase the mutation rate 10 to 1,000-fold. That means the mutation rate can be, instead of 1 times 10 to the 6 replicated genes, it can be from 1 in 1,000 genes to 1 in 10 to the 5th genes. Any question about any of that? Identifying mutants. You can test for mutants by a positive or direct selection or a negative indirect selection. If this were to go walking down the street, people would say, yep, there goes a mutant. And that's because you can tell that this is not a normal individual. And you can detect that just because of the way the individual grows or appears different from a normal human. So that's positive direct selection. Negative indirect selection detects mutant cells because they do not grow. Let me talk about negative selection a little bit in greater detail. I actually did this when I was an undergraduate, looking for mutants by negative selection. And that is, you have a plate here, which has individual isolated colonies, and you can take a stamp of velvet and place the velvet onto the colonies. The colonies will then put cells on the velvet, and then you can stamp the velvet on 2 other plates. This one has a plate containing media containing the amino acid histidine. This one, the media lacks the amino acid histidine. And then we grow up the plates to look at the colonies. And this one, move that up here now. This one has all the colonies of this plate. So right there, I think is that stamp there. And that one we see is, which one is that? That one there. This colony here grows on the media containing the amino acid histidine, but here, that colony is not growing. So these cells are mutant, and you must supply the amino acid histidine, or the cells will not grow. Any question about any of that? Anyways, that's negative selection for looking at cells that cannot grow because they are a mutant. All right, let's change gears a little bit and talk about genetic recombination. Recombination is the exchange of genes between two different DNA molecules. It often involves crossing over, which happens in mitosis and meiosis, at least in eukaryote cells, where let's see, where do I have that there? I don't have it labeled here. Where the mother's chromosome with the lowercase alphabet alleles pairs up with the father's chromosomes, which have the uppercase alleles. And then the DNA molecules actually cross over and then break right here. And then the mother's chromosome right there now has part of the father's chromosome. And then in vice versa, the father's chromosome is part of the mother's chromosome. Crossing over happens, like I said, all the time in mitosis and in meiosis of eukaryotic cells. What's the difference between recombination and crossing over? Are they the same? Anyone have a guess? Crossing over is a little more specific. All crossing overs involve recombination, but not all recombinations involve crossing over. For example, suppose we had a chromosome, which part of the genes move from this part of the chromosome to this part of the chromosome. That's not crossing over, but it is recombination. Exchange of genes between, actually in this case is one DNA molecule, but we could also have a portion of this chromosome move to that chromosome with no portion of this chromosome moving to that chromosome. We've seen that every once in a while, like when the Y chromosome gains a little bit of the X chromosome. It's very rare, but it does happen. And there's other examples. I'm just giving a few examples. So recombination is exchange of genes between, I guess, one or two more chromosomes. And recombination is when two DNA molecules pair up, break, and then exchange pieces. Recombination, let me back up just a minute, is important because it allows new gene combinations to occur. For example, here we have the lowercase g now associating with the uppercase H gene. And before we only had lowercase g with the lowercase h and the uppercase G associating with the uppercase H. So this is a new genetic combination. Any question about any of that? What did I say? It reshuffles the genes in the deck. a sexually reproducing species actually reshuffles the deck because two parents make one offspring. But crossing over and recombination increases the shuffling of the genes in the deck of the individual. Let's talk about genetic transfer. Vertical gene transfer occurs during reproduction between generations of cells. So when one parent cell divides into two offspring cells, or for humans, two parents give rise to one offspring. That's vertical gene transfer. Horizontal gene transfer is the transfer of genes between cells of the same generation, where DNA leaves the donor cell and then that DNA enters the recipient cell. Horizontal gene transfer can happen in one of three ways: transformation, conjugation, and transduction. Transformation is where DNA leaves one cell. In the natural environment, that cell probably dies, the DNA leaves the environment, and then that DNA can be taken up by the recipient cell. I'm not going to go into it, but the recipient cell takes up the DNA, and then the DNA has to be brought into the chromosome. This chromosome now has a new gene that was not in this cell before, the lowercase a gene that came from the naked DNA in transformation from another cell. This is important, like if lowercase a codes for antibiotic resistance, this cell now gains antibiotic resistance. I will talk about why that's important in just a little bit. Here's a case of transformation where this cell sees a piece of DNA there, takes up the DNA, has indigestion as that DNA becomes part of the cell, and then it expresses new genes which can change the phenotype of this cell, going from this to, I don't know, that cell. Conjugation, also called sexual conjugation, happens when DNA leaves one cell and enters another cell. It can happen in one of two ways. In a gram-negative cell, we have the sex pilus, which is made by this cell, and the sex pilus is hollow. The plasmid in this cell can move through the sex pilus and then join the F minus cell. The F plus is called F plus because it has the F plasmid in it, which codes for the sex pilus. And then both cells, because the F plasmid replicates here, and then one copy is sent across the sex pilus to the F minus cell. That F minus cell now has the F plasmid in it, which codes for the sex pilus, and that'll change the F minus cell to an F plus cell. So this is the way that conjugation happens in a gram-negative cell. In a gram-positive cell, we can have sexual conjugation happening by a mating bridge, where two gram-positive cells come together. They then lose the cell wall right here and here. And then the cell membrane of this cell actually fuses to the cell membrane of this cell. And then it opens up, allowing the cytoplasm of this cell to exchange with the cytoplasm of that cell, allowing plasmid and DNA to move from this cell into that cell and vice versa, from this cell into that cell. Anyways, that's how sexual conjugation can happen in a gram-positive cell. Any question about any of that? The Sex pilus. Let me go back a minute. The sex pilus coming out of this cell literally skewers this cell and then acts like a winch to bring these two cells together. They do not have to be brought together like this, though, because the sex pilus is hollow. And even if they're at this distance, the plasmid can move through the sex pilus and then move into the F- cell. As you can see, the plasmid replicates here, and then one copy is sent into the F- cell. The F- cell now has the F plasmid in it. The F plasmid codes for the creation of the sex pilus. So this cell is now the F plus cell, as well as that one, because it has the F plasmid in it, and it will give rise to a sex pilus. Any question about sexual conjugation? Transduction happens with a virus. Normally, a virus will bind to the cell wall of a cell and inject its DNA or contents into the cytoplasm. The first thing the viral DNA often does is degrade the host cell's DNA. Why does the virus DNA degrade the host cell DNA? Because the virus DNA wants to call the shots in this cell. And an easy way of calling all the shots in the cell is to degrade the host cell's DNA. Now your DNA is going to determine what happens in this cell. And then the viral DNA will replicate itself, making multiple copies. And then the viral DNA will be expressed, making viral messenger RNA, which in this case is already in the cytoplasm. And then the viral messenger RNA will combine with the cell's ribosomes and the cell's tRNA to make the viral proteins, which will make the head of the virus, which will then get together with the viral replicated DNA to make a new virion. And then the last thing the virus does is degrade the cell wall, so the cell will burst, releasing the new virions, the new viruses, which will then go out of this cell and bind to a new cell. But viruses are not terribly worried if they make a mistake because they have lots of offspring. Let's take a look at this viral DNA or that viral DNA. And you'll note that it's not the red-purple viral DNA. What it did was it put in a piece of the cell's DNA into the virus. When this cell now binds to another cell and injects its contents into the cell, that's not viral DNA, which is being added to this cell. That is the DNA of this cell. That DNA can then combine with the host chromosome, and now this cell has new DNA that came from this cell. Any question about any of that? Why is that important? Well, let's say that this piece of DNA, which came from right here, has the gene on it for antibiotic resistance. This cell now has gotten the gene for antibiotic resistance. Any question about any of that? All right, let me stop the show here. Come on. So, transformation, conjugation, transduction, and then let's come up here and go spontaneous mutation. Oop. Spontaneous mutation happens about 1 out of every 10 to the 6th cells. Transformation happens about 1 in every 10 to the 5th cells. And that's true for conjugation in bacteria. And that's true for transduction in bacteria. Meaning transformation, conjugation, and transduction are 10 times more common than spontaneous mutation. Showing you the importance of transformation, conjugation, and transduction. And that means, like, once we have a patient in the hospital which has a gene for antibiotic resistance, it's true this can come about by spontaneous mutation, but that would be 1 in a million cells that would get it. And it will be coming about by transformation, conjugation, and transduction 10 times more frequently than spontaneous mutation. Showing you the importance of transformation, conjugation, and transduction. Any questions about any of that? All right. Plasmids are small circular extrachromosomal DNA. So they're a part of the DNA which are not part of the chromosome. They do contain genes. What differs is there's only a few genes on a plasmid. Like here's a plasmid. It might have 5 or 6 genes on it, maybe 7. I didn't really count. And that's very few compared to a chromosome, which may have 20 to 40,000 genes on it. And it depends on the size of the chromosome. Like the Y chromosome has much fewer, the chromosome 23 has much fewer. And I guess I was thinking more in the terms of a chromosome of a bacteria. Plasmids are important, though, because a plasmid is more likely to exchange between cells than the chromosome. Plasmids also replicate independently from the cell's chromosome. And we actually already saw that right here. This plasmid replicated independently of the chromosome. This plasmid replicated here, the chromosome did not. Any question about any of that? There are different categories of plasmids. That's all you need to know. You don't need to know the names and the types of different categories. Transposons are a piece of DNA that moves from one region of the DNA to another region of the DNA. In eukaryotes, a transposon can move a piece of DNA from one chromosome to another chromosome, as well as it can move DNA from one chromosome to another region of that same chromosome. Transposons always have the transposase gene in the middle of the transposon, and they have inverted repeats on the ends of the transposon. It's believed that the inverted repeats help the transposon integrate into a new piece of DNA, as well as it may help the transposon come out of the piece of DNA it's in. The transposase gene is something that actually cuts the transposon out of the piece of DNA, and then it goes to another region of DNA. The transposase gene cuts that DNA, and then the transposon inserts itself into that cut piece of DNA. Any questions about a simple transposon? Shown here. A complex transposon can carry other genes on the transposon, like TN5 carries the kanamycin resistance gene, and a complex transposon usually has the extra DNA in between two transposable units, meaning there we have transposase, there's an inverted repeat, there's an inverted repeat, there's another transposase, and there's an inverted repeat and an inverted repeat. So the complex transposons usually is a transposon that has a simple transposon on each end of the larger transposon. Does that make sense? Hopefully it did. The importance of a transposon is that, well, first of all, they reshuffle the DNA, but also they can allow a piece of the chromosome to then integrate into a plasmid. So there's the transposon. It moves from the chromosome into the plasmid, or vice versa. And it's important for plasmids because plasmids are more likely to leave this cell than the chromosome is likely to leave the cell. Any questions about any of that? So transposons sort of act similar to recombination, and that is they tend to shuffle up the genes in a species and allow different combinations of genes to be associated with each other. All right, any questions about the genetic material that we've discussed so far? If not, let me briefly talk about genes and evolution. Genetic variation results from, if it's new genetic variation, can only happen by either spontaneous mutation or mutagen-induced mutation, meaning if the gene is not found in the human population, the only way it can enter the human population is by spontaneous mutation or, I guess, a mutagen-induced mutation. And we gave an example of that 10,000 years ago. Humans only had brownish-eyed alleles. And 10,000 years ago, we had a spontaneous mutation in Northern Europe where the blue-eyed allele came into existence. But once the gene exists in a population or in a species, The gene can move between individuals by transformation, conjugation, transduction, transposition, although transposition moves the gene from a chromosome to a plasmid, and then the plasmid is more likely to move by transformation, conjugation, or transduction out of one cell and into another cell. Crossing over, like transposition, doesn't create new genetic variation, but it creates new variations of gene combinations where you can have like the uppercase G allele associating with the lowercase h allele. And previously, only the uppercase G allele was with the uppercase H allele, and then vice versa. The lowercase g was only associating with the lowercase h allele. So crossing over can shuffle those genes, mix them up. Transposition increases that. And then once the genetic variation occurs in the population, it can move around by transformation, conjugation, or transduction, as well as it can be created by mutation. All of this genetic variation drives evolution, and how evolution occurs is by natural selection. Any questions about genes and evolution? Any questions about Chapter 8? All right, that's it for Chapter 8. Let's move on to our next chapter, which is, I think, Chapter 14, but let me check. Nope, Chapter 13. Viruses, viroids, and prions. Let me shut that down. So as normal, I give you the chapter 13 discussing viruses, viroids, and prions. I give you the major terms. What's the capsid to a virus? What's an envelope? What's a spike to a virus? What's a bacteriophage? What's a lytic virus? What's a lysogenic virus? What's a prophage? What's a provirus? What is a latent infection or virus? What is a persistent infection? What are prions? And what are viroids? So these are the major terms. And then 2, understand the characteristics of viruses, their structure, and their classification. 3, understand about viral multiplication. And then 4, understand about prions and viroids. Any questions about what we're going to cover? All right. The only goal a virus has is to replicate. And how do viruses replicate? Well, they infect a host cell, take over the control of the host cell, and then use the host cell to make viral copies, which then escape, and the new viral replication cycle can continue. Any questions about viruses so far? Let's watch a little video of virus replication. Phage is another term for bacteriophage, also called phage.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, These viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any questions about that? So some characteristics of viruses. Viruses are inert when they are not within their host cell. Viruses display little or no metabolic activity when they're outside their host cell. So viruses only have metabolic activity when they are inside their host cell. Viruses require a living host cell to multiply, so we can say that viruses are obligate intracellular parasites, a parasite that requires a cell in order for the parasite to replicate. Viruses within their living host cell use their host cell enzymes, nucleic acids, amino acids, tRNA, ribosomes, etc. to replicate the virus. Viruses have no ATP generating mechanism. They only use the ATP within their host cell. Viruses can contain DNA or RNA for their genome. They never have both DNA and RNA. It's either a DNA genome or an RNA genome. A DNA genome is normal because that's the genome of all cells. But viruses, their genome can be RNA, which is unheard of in a cell. Viruses always have a protein coat surrounding the genome. Here we have the protein coat, this section cut out, so you can see the innermost molecule, which is the genome, either DNA or RNA, and then the protein coat surrounding the genome. Some viruses are also surrounded by an outer lipid envelope, which is outside the protein coat. So it would be about in this region. Not all viruses have an envelope. The host range of a virus is the range of host cells the virus can infect and replicate in. This is determined by host-specific attachment sites. We call that attachment site a host cell receptor that the virus binds to. And then the virus has molecules which bind to the host cell receptor. Let me blow this up. And these are called ligands or adhesions, the molecule on the virus that binds to the host cell receptor. Most viruses only infect the cells of one or a few species, and usually they do not infect all the cells of one species. Like HIV can only infect the cells of certain cells of humans and cells of one of the chimpanzees. All other species of animal, the virus cannot infect and replicate in those cells. Is that clear? Oh, let's talk about the cells that the HIV can infect in human cells and replicate in. Obviously, T cells, monocytes, and actually monocytes, macrophages. It can also bind to and replicate in brain cells and a few other cells in the human body. It cannot bind to and replicate in skin cells, muscle cells, probably bone marrow cells. Most of the cells of human, HIV cannot replicate, bind to and replicate in. Any question about any of that? And this host range is determined by whether the cell has the correct receptors which bind to the molecules on the virus that we either call ligands or adhesions, depending on your author. Now, there are some exceptions, like Eastern equine encephalitis. It can replicate in human brain cells, maybe some other human cells, and horse cells, at least the brain cells, and probably some other horse cells. I think pig and a few other mammals, like rodents, donkeys. I'm not sure of all the mammals, but at least those. And it can also infect and grow in bird cells, many different species of birds, and it's transmitted by mosquitoes. I don't know if the virus actually replicates in mosquitoes, though. So that virus can replicate in many different cells of many different species. But most viruses are rather particular, infecting only a few host cells of one or a few species. And HIV is a good example. Any question about that? All right, bacterial viruses are called bacteriophages or phages, and sometimes they're just called phages, but I will always use the term bacteriophage because I want to emphasize that this virus is infecting a bacteria. Any questions about any of them? All right, here we're looking at the relative size of viruses. One of the smallest viruses that you are familiar with is the poliovirus. There are viruses a little smaller, but nobody's probably ever heard of bacteriophage MS2. At 24 nanometers, poliovirus is only 30 nanometers. And here we have the different sizes of viruses, including this one, megavirus, one of the biggest viruses around by volume. The Ebola virus is actually longer than megavirus, but by its total size, if we go by its volume, the Ebola is smaller than megavirus. Any questions about any of that? And there we have a bacterium, an average bacterium, E. coli, compared to the size of a red blood cell, much larger than the prokaryote cell, much larger than most of the viruses. I mean, at least twice this one and that one. Any question about any of that? So the very biggest virus is about 440 nanometers in diameter. That would be megavirus. And about the smallest viable cell is about 400 nanometers. So the viruses do get up to the size of the smallest bacteria. But generally, viruses are about 1/10 the size of an average bacteria. Any question about any of that? All right. So let's talk about viral structure. A virion is a single mature, complete infectious virus particle. So when a virus replicates, we say the replicated viruses are the new virions. Is that clear? The genome of a virus can be either DNA or RNA. If it's DNA, it can be double-stranded, and that's what most of the DNA in our cells is normally. But the viral genome can be single-stranded. And this is heard of in eukaryote cells as well as bacteria cells. And that is whenever the DNA is replicating or whenever the DNA is being transcribed, the DNA is single-stranded. Any question about any of that? So viruses can be double-stranded DNA or single-stranded DNA. They can also be RNA, and if they're RNA, they can be double-stranded RNA. Double-stranded RNA is unheard of in cells. We do not have double-stranded DNA in our cells. The RNA genome can also be single-stranded, and if it is single-stranded, it's important to mention, is it positive single-strand RNA or negative single-strand RNA? If it's positive single-strand RNA, this positive single-strand RNA can be read by the ribosomes the same as messenger RNA. And that's because the positive single-strand RNA code for all of the nucleotides that the the virus needs. And it'll have AUG on it, etc. If the viral genome is, on the other hand, negative-stranded single-strand RNA, this RNA genome cannot be read by the ribosome. It is complementary to the positive single-strand RNA. So, for example, in the positive single-strand RNA, we'll have A, U, G, which is important in telling the ribosome to begin protein translation at AUG. In the negative single-strand RNA, we do not have AUG. Let me figure this out. Instead, we'll have U, AU, so it'd be A, and then C. And the negative single-strand RNA cannot be read by the ribosome. Any question about any of that? All right. The viral genome can be linear or circular. A linear viral genome, at least linear DNA, would be similar to our human DNA, meaning all of our DNA is in the chromosomes, so the DNA molecule, like on the X chromosome, it begins here at that X, and then it ends here at the bottom of that X. And some viral genomes are that way. Some viral genomes, on the other hand, are circular, forming a circle where the beginning point is the same as the ending point. And most bacteria have circular DNA. Some viruses can furthermore have their genome in several segments, like influenza virus has their genome in several segments. I used to know how many segments, but I've forgotten. It's something like 6, but don't quote me on that because I don't remember how many segments in influenza virus. Having the genome in segments should not be new to you because humans have their DNA in segments. It's true we don't call them segments. We call them, I just lost what the heck we call that. I had it on the tip of my tongue and now I've forgotten. What do we call? Oh, we call the several segments of DNA in a human, we call that the human chromosomes. Any question about any of that? All right. So we mentioned that all viruses have a protein coat on the outside of the virus. The protein coat is composed of subunits called capsomeres. Let me blow this up. So this virus here has one capsomere there, and the only thing making up the protein coat is that one capsomere, just repeating many times to make the viral protein coat. Viruses can have more than one capsomere. Let me show you a virus having one more than, excuse me, more than one capsomere. Well, I didn't see it. So we'll just use this. The head of the virus, this portion here, contains the genome, and the head is composed of one capsomere, but the sheath has either one or two capsomeres. The base plate has one or two other capsomeres, and then the tail fibers probably have a few capsomeres. At least one, but probably a few. See where we were. So the capsomere can be only one making up the protein coat, but the protein coat could be made up of several capsomeres. If we have a virus that only has the what was that called? If it only has the protein coat and there's no envelope, it is called a naked virus. So if the virus has no envelope, we call it a naked virus. If the virus has an envelope, we call it an enveloped virus. The envelope of a virus is a lipid bilayer. It does have proteins and carbohydrates in it, but the main molecule will be the phospholipid molecule. The envelope may have some virus proteins in it or glycoproteins. Let me blow this slide up. And then if the virus has an envelope, it may have glycoprotein spikes, which insert out of the envelope. The glycoprotein spikes are important to both the cell and then the host that this virus infects, because the glycoprotein spikes are the outermost molecule on the virus. And so if the virus has spikes, the outermost molecules will be in the spikes. The spikes serve as attachment points of the virus to the host cell receptor. So that means the spike has the ligands or adhesions for binding to the host cell receptor. And then the spikes are also antigens, which the animal who becomes infected with this virus, and if the animal mounts an immune response, much of those antibodies, if not all of them, will be binding to the spikes. And why is that? Because antibodies bind to the outermost molecule on a cell or a virus, and the outermost molecules are the spikes. So the spikes, the glycoprotein spikes, are important in helping the virus bind to its host cell receptors, as well as it helps the cell get rid of cells which don't help you with anything because they're not one you need. Any questions about any of that? All right, let me check the time here. Oh, we got time. So once we're talking about the viral structure, it's important to talk about the viral morphology. The morphology is the shape of the virus. Viruses are classified on the basis of their capsid structure, meaning their shape. And we have three main categories of viruses. We have a helical virus. Let me blow this up. Where the capsid is in a helix. Right there. I don't see the helix in this, so if somebody sees it, they can point it out to me. That's the first, the helical virus, where the capsid is in a helix. There's also a polyhedral virus, where the virus has multiple sides. This sort of looks like a complex dice with multiple sides, much more than 6. I don't know how many there are. 1, 2, 3, 4, 5, 6. This is at least 12-sided, maybe up to 20-sided sides on the virus. So helical and polyhedral are pretty straightforward. We also have what we call complex viruses. These are viruses that have a complex shape. And any virus which is not helical, not polyhedral, is a complex virus. Bacteriophage T4, which looks like the lunar lander, is a complex virus. But so is this virus here. And actually, that's kind of close to T4. Let me see if I can read that. Oop. Oh, that's a pox virus. Probably not smallpox, but one of the pox viruses. Any questions about the morphology of the virus shape? Let's talk a little bit more about bacteriophage T4. T4 is a bacteriophage, a virus that infects bacteria, but belongs to the T-even family, meaning T2, T4, T6. T4 is important because that is the virus that has been studied more than any other virus. T4 is a bacteriophage that can infect E. coli. And I suspect that's why T4 was the virus that microbiologists decided to study the most. And it's simply because microbiologists had E. coli on hand in many microbiology labs. And when they started studying viruses, they happened to choose T4 to be the virus that they studied. Here it is there. There's the head of T4 containing the DNA genome. The sheath here containing at least 2 segments of material. The base plate here also containing at least 2 segments. and then the tail fibers here. The tail fibers are important for bacteriophage T4 because it's this portion of the virus that contains the adhesion molecules or the ligands that bind to the host cell receptors. Any question about any of that? So remember, bacteriophage T4 is the virus we know the most because it's been studied more than any other virus, including a human virus. All right, what happens when a virus infects a host cell and then the host immune system responds to that? Infection with a virus in an animal can stimulate the host to produce antibodies. Antibodies are a part of the human immune response, meaning the immune cells, at least some of them, make the antibodies, which then go out and bind to the virus. Antibodies recognize and bind to the viral surface proteins. We call these viral surface molecules antigens. What the host is responding to are certain molecules on the infective agent that we call antigens. And as I mentioned before, if the virus has spikes, then the spikes are the viral antigens. So the antibodies would recognize and bind to the spikes. Some viral proteins can change or mutate frequently. Therefore, different strains can come about, and different strains can infect the same individual more than once. Normally, when a human is infected with a virus, the host immune system will respond, and that will create antibodies and other host defenses to respond to the virus and then start attacking the virus. But if you got infected with one strain of the virus and this virus mutates or changes frequently, then another strain of host cell would not be sensitive to that antibiotic. Any question about any of that? All right, an example of a virus that changes or mutates frequently include influenza virus, the flu. And that's one reason why I mention that if you come down with the flu, you should get a new flu shot about every 3 years. Let me see if I can get this going. There we go. And the reason is if we have the percent immunity on the x-axis and then the time on the y-axis, let me go ahead and put that in. And on the y-axis, we have percent immunity. So up here would be like 100%. You never quite get a virus that will, 100% of them will react. So there's like 50%. 25%. So with immunity, the immune response weakens with time. Something like that. Oop, not very straight, but you're getting the point. And after 3 years, your immunity has decreased to something like that. And they say at this point, you're no longer protected by the vaccine. You're never at 100%. So let's make 100% up here. But it's quite high initially after you get the infection or you get the vaccine for the infection. Like H1N1 or the flu virus, if you got the flu shot, then within the first year, your immunity is still pretty high. It's not what it used to be. Maybe this is 70%. Let's go 90%, which is a bit high. And then this would be something like 75%. Let me erase that now. Who's getting in there? Oh. And over time, the immunity will decrease. So 2 years about here. Oh, come on. Oh, I got it colored white for some reason. So 2 years about there. And 3 years someplace around here. After 3 years, the flu shot will no longer protect you from the mutated strains of the virus because the virus mutates that rapidly that after 3 years, the mutations will be different enough that the original flu shot will no longer protect you. And generally, they want immunity to be high, so they recommend getting a flu shot every year. Any questions about any of that? All right, so the flu is a virus which can change or mutate frequently. And like I said, after 3 years, you should get a new flu shot because your protection from the original flu shot will be gone. What's another virus that mutates rapidly? And so you need to get a vaccine fairly frequently. Anyone have an example?
COVID.
COVID-19 is a good one, yes. That one, you probably need to get a new COVID vaccine about every year because new strains come out all the time. About every 6 months, we have a new strain of COVID-19 coming out, which is a little less than the flu, but it's similar. Unfortunately, most viruses do not mutate that frequently. For example, we are still giving the 2 polio vaccines to protect people from the disease polio. And these vaccines were made in the 1950s, and they are still working today because polio does not change or mutate frequently. And there are many vaccines that are still in use today that were made a long time ago. I mean, if it was 1950, which it wasn't, it was something like 56 or 58. There's 2 polio vaccines, and I don't know which one was made in which year, but the point is they were made in the 1950s, so that's almost 75 years ago. And we have older vaccines than that, like the BCG vaccine, I think was made in the early 1900s. Any question about any of that? All right. Virus taxonomy is the grouping of virus into families based on the different characteristics the virus have. You'll be pleased to know that, let's see, what's above family? Orders. Viruses have an order, and then they have families, they have genuses, they have viral species, and then subspecies. But higher than an order, most viruses do not have. We do not lump viruses into phyla, subphyla, classes. Obviously, we do have orders, and then family. And the reason being is that viruses are typically small in their genome, and then the virus also changes rapidly to mold to the host cell that they're infecting. So we just do not know the evolutionary history of viruses because their genome are so small, and then they also change very rapidly to adapt to their host cell. There is one exception, though. Viruses do not have a domain because they're not cellular and their genomes are too small also. But we do not separate viruses into domains, but we do separate viruses into the kingdom that they infect. So we have plant viruses, we have animal viruses, and we have bacterial phage viruses that are called bacteriophages. But the other higher orders we just do not have. We don't know what they are. And you have to come down to right before families before we can classify viruses. Any question about any of that? All right. Viruses are always grouped into families. We base the family on the type of nucleic acid the virus has, the replication strategy, and the viral morphology. A viral family name always ends in viridae. The genus name of the virus always ends in virus. The virus species is a virus that is grouped by sharing the same genetic information and the same ecological niche, meaning a species is a virus that infects the same viral host cells. Like HIV, one is a species which always infects the same host cells, ecological niche, and then the same genetic information. You'll be pleased to know that virus species only have common English names. Like poliovirus, that is its scientific name, poliovirus. And all virus species use the common English names. It turned out that the first virologists were all English speaking, and then they gave viruses their names. And then the other people who began studying viruses who spoke a different language, they used the same names that the English speakers already gave to them. So if ever you want to talk about polio or poliovirus, wherever you go in the world, you simply use the poliovirus. Does that make you happy? You don't need to know any Latin scientific names. All you need to know is the common English name. I think somebody said yes. All right. A subspecies is the same name of the virus species, and then you put a number by it. Like there is human poliovirus 1, called poliovirus 1, and then there's poliovirus 2. The subspecies is very important for us because poliovirus type 1 is the virus that causes the disease that we call polio. Poliovirus type 2 is the virus that we use as the live virus vaccine that we use to protect people from getting the disease polio. And that shows you the difference of the subspecies. Any question about any of that? All right. Here's viral taxonomy. The family is the herpes family of virus, and we call that herpes viridae. The genus of one of the herpes viruses is the simplex virus, and that is the genus name. The species would be the oh gosh, what the heck is that? Herpes simplex virus. Any question about that? And then the subspecies is herpes simplex type 1 or herpes simplex type 2. They share similar genetic information. They can infect the same cells, although herpes simplex type 2 tends to infect genital cells, causing a sexually transmitted genital disease. Herpes simplex type 1 tends to infect the mouth or oral region, causing what we call cold sores. Any question about any of that? You'll note that I'm calling it the simplex virus rather than the human herpes virus, because this human herpes virus, meaning the herpes simplex virus, is the name that clinicians and the general public has stuck to using. The human herpes virus is a scientific name for herpes simplex virus, and herpes simplex continues to be the name that the majority of the people, including clinicians, use. So I no longer even talk about human herpes virus. Any question about any of that? All right. Bacteriophages can be grown in the lab, and I used to grow them up this way, where what you would do is you'd have a lawn of bacteria growing on the plate. Let me blow this up. That's why the plate is off-white in color, from the lawn of bacteria growing everywhere. And once you plate the bacteria before it grows up to form the lawn, you then put on a solution containing a virus. Wherever the virus binds to a bacteria, the virus will infect the cell and then replicate. And where will that, and the virus will then lyse the cell to release the newly replicated viruses. And then where will those viruses go? They'll go to nearby bacteria. And so you get enough of this happening that you'll get a region here. Let me blow that up even bigger. You'll get a region here where we'll have a plaque. And that's because all of the bacteria in this plaque have been lysed by the viruses. So it started with one cell getting infected, the virus replicated, the virus went to neighboring cells, lysing those cells, infecting those cells, releasing more cells, more viruses, infecting more viruses, cells around it, and then you get a plaque. You can count the number of viruses on the dish here to count the number of viruses in your initial excuse me here, in your initial solution. You can notice that this is a much larger plaque than this one there. Why that might be is simply because the larger plaque, we had a cell here, and a virus initially infected that cell at the very beginning. Whereas where we have a smaller plaque over here, we have a virus right there, and we have a bacteria cell here. We needed that bacteria to replicate before let's go ahead and replicate it one more time. Before the virus could infect the bacteria cell. Any question about any of that? So the larger plaques, let me blow that up again. The larger plaques here, the virus may have infected the cell at the very start. And then the virus here probably didn't infect the virus, it infect the bacteria cell until the bacteria cell grew to eventually touch the virus. Now, some of the plaques, like this one here, is apparent, let me blow that up, it's apparent that this is 3 plaques that fuse together. One here, one there, and one there. But that one there looks like it's 1 plaque. Any question about any of that? Anyways, you can grow the viruses up and then count the number of viruses in your original solution by counting all the plaques. And this would be a lot, over 100, maybe even over 200, but less than 250. So remember, in viral multiplication, the viral genome injects itself into the bacteria, which then hijacks the cell. The viral genome only contains a few of the genes needed for new virion synthesis, but the virus uses the host cell's machinery to replicate more viruses, meaning the virus uses the host cell's ribosomes, host cell's tRNA, host cell amino acids, some of the host cell enzymes, ATP from the host cell. The basic mechanism of viral multiplication is similar in all viruses. And that is the virus binds to the cell, gets inside the cell, takes over the machinery of the cell to replicate new viruses. There are 2 viral life cycles that bacteriophages use to replicate the virus. There's the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the virus replicates and it will lyse the host cell. So the host cell is killed, and when the host cell is lysed, that releases the newly replicated virions. The lysogenic life cycle, the host cell remains alive for as long as the virus remains in the lysogenic life cycle. Any question about the 2 life cycles that bacteriophages engage in? All right, So here's a question for you. To make a new bacteriophage, what do we need? What components does the virus need to synthesize to make a new bacteriophage? There's two. All viruses have two, at least two. All right. To replicate a virus, if we have a double-stranded DNA virus, we have to replicate the viral genome. Doesn't matter what the viral genome is, you have to replicate the viral genome. And you must transcribe that genome to get viral messenger RNA, to get viral proteins. Once the viral proteins are made, it'll make the capsid proteins, making the coat of the virus. You must get the capsid proteins together with the replicated viral genome to make the new progeny virions. So these two things always need to be replicated. The viral genome and then the viral proteins. With the viral lytic life cycle, the lytic life cycle follows 5 stages in the life of the virus. A life cycle means the cycle the virus takes to live its life. So the life cycle is how the virus infects the cell, replicates, then releases itself to go on and infect another cell. And they always follow, in the lytic life cycle, they always have attachment where the virus attaches to the host cell, then penetration where the virus gets inside the host cell, and usually it's only the viral genome that gets inside the host cell. And then there's biosynthesis, where the virus makes new replicated copies of the viral genome, as well as makes the proteins, the viral proteins of the protein coat. And then there's maturation. That's where you assemble the new phage particles, and then you get the newly replicated virions. And then there's release, where the virus releases itself from the host cell. And in the lytic life cycle, that happens by the virus secreting the enzyme lysozyme, which breaks down the cell wall. Water will then enter the cell until enough water enters the cell that the cytoplasm bursts, and that will release the newly replicated viruses. Any question about any of that? If not, let's watch a little movie on the lytic life cycle.
First, the phage.
Sorry, my computer doesn't have a program to read this movie, so let me start the first part of the next movie. Let's see if I can get there.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about that? So let's go on in a little more detail here. First, the virus attaches to the host cell. I guess I only have it here. The virus then contracts. Well, actually, the virus releases a little bit of, let me blow this up. Right there. The virus releases a little bit of lysozyme, which digests the cell wall right here. And then the virus contracts, which is kind of shown here. That puts pressure on the head, and that will inject the contents of the head into the cell, which in the movie was shown, if I can find it. Nope, that's wrong.
Cell's enzymes and organelles to attach to a certain type of bacteria. Next, the phage breaks down
All right, so that happens by a little bit of lysozyme being released, which digests the cell wall.
the cell wall, and the nucleic acid from this capsule
And then the tail contracts, and that puts pressure on the contents of the head, which injects the contents of the head, the viral genome, into the cell. Any question about any of that? Let's go to where we were about.
reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
The lytic cycle or the lytic life cycle. All right, now the viral genome is inside the cell. One of the first things the virus does is it secretes an enzyme which actually digests the host cell chromosome. And that's because the virus wants to call the shots in the cell. And now the cell is being controlled by the viral genome. The viral genome will use the host cells, and that's called penetration, getting the virus into the viral genome into the host cell. Then we have biosynthesis, where the virus replicates the newly replicated viral genome, as well as the proteins of the virus. So we have to make the heads, and then the sheath, and then the tail fibers, as well as the viral genome, which I don't see here. Well, maybe it's already made inside the head, meaning it's already stuffed inside the head. And then we have maturation, where the viruses come together, and the virus parts actually self-assemble. There is an enzyme which stuffs the viral genome into the head, although it could make a mistake and add the a piece of the host cell's DNA, but most of the time it's the viral genome that gets stuffed into the head. And then one of the last things the virus does is it codes for the enzyme lysozyme, which is released, and that attacks the cell wall, digesting it, and then water will move into the cell, eventually causing the cell to burst. And that will release the newly replicated virions. Any question about any of that? Let me see if I went through all of them. Attachment, penetration, biosynthesis, maturation, the releasing of the newly replicated virions is called the release. There are two things you should know about the lytic life cycle, and that is the burst time. That's the time from initial infection of the host cell to the time that the host cell bursts, releasing the newly replicated virions. That averages 20 to 40 minutes. It will depend on the species, as well as the host cell and the temperature that you're growing everything at. And then the burst size, that's the number of newly released virions. It's usually between 50 and 200 newly replicated virions released from a host cell. Any question about any of that? All right, that's the lytic life cycle. Let's talk a little bit about the lysogenic life cycle. When a virus follows the lysogenic life cycle, meaning it's a lysogenic virus, when this virus initially infects a bacteriophage, it must decide immediately to do one of two things. It can, one, enter the lytic life cycle, or two, it can choose to enter the lysogenic life cycle. If the lysogenic virus enters the lytic life cycle, it follows this path here, which I've already discussed, the lytic life cycle. The viral genome is injected into the cell, stays independent, the viral genome, and then it replicates more viral genome, replicates the viral proteins, which are assembled into new virions, which causes the cell to break by secreting the enzyme lysozyme, releasing the newly replicated viruses. And that's the lytic life cycle, the same as what we talked before. However, a lysogenic virus, once it is injected into the cell, can choose to enter the lysogenic life cycle, in which case it follows this cycle, not that cycle. In the lysogenic life cycle, the viral genome integrates into the host cell chromosome, and that we call a prophage, where the viral genome is inside the host cell's chromosome. Now, the only time the virus replicates is when the host cell replicates. So when the viral DNA replicates, you also replicate the viral DNA. So when the host cell DNA replicates, the viral DNA replicates. And this can go on for many cell divisions, where the virus continues to stay in the lysogenic life cycle. However, every once in a while, the virus can leave the lysogenic life cycle, and if it does, the prophage will be excised from the host cell's chromosome and then become independent of the host cell's chromosome, and then the virus will enter the lytic life cycle, and that will kill this cell. Any question about any of that? All right, let's watch the rest of the movie, which talks about the lysogenic life cycle. Actually, I've only got 1 more minute. I'm not going to show you the movie. I'll show you the movie next time. Any questions about that? All right, I'll see you actually in the lab. There is no lab today. I'll be there to log in, just to answer questions for 15 minutes. But after 7:15, I'm going to log out. So if you have a question in the lab, I will be there from 7 to 7:15. All right, any questions?
Can you show slide 22 just real quick so I can copy it?
Sure. There you go.
Thank you.
You're welcome. All right. I'll say goodbye, unless there's other questions.
Thank you, Professor. Have a good night.
All right, you too. Bye.
Goodbye.